Okay, in this video, let's take a look at cholesterol metabolism. So cholesterol comes from two sources. Let's look at them individually. First source is from dietary. Second source is from endogenous synthesis. And in this case, we're going to be talking primarily about the liver because that is the organ that synthesizes the greatest amount of cholesterol. Now, dietary cholesterol, recall from our digestive lectures, is in the form of an ester. So it's a cholesterol ester. What that means is that we have a fatty acid that is covalently bound to the 3' the 3-carbon hydroxyl group on cholesterol, creating the cholesterol ester linkage. In our gut, we have pancreatic cholesterol esterase enzyme secreted by the pancreas during the digestive phase, the intestinal phase, and this will break up the cholesterol ester into cholesterol plus free fatty acid. The free fatty acid is, of course, recall that it is taken up by the intestinal enterocyte by fat P, which is the fatty acid transport protein. Cholesterol we haven't talked about yet, and this is taken up by a cell surface transporter that is abbreviated NPC1L1 for the Niemann-Pick C1 like 1 protein. This particular protein, this form of the Niemann-Pick protein, is localized on the apical surface of the intestinal enterocytes and responsible for uptake of dietary cholesterol. There is another Niemann-Pick protein that many of you may be aware of. That is simply called the NPC1 transporter. This is involved in cholesterol transport in cells. Both of these proteins have structural similarity that they can bind and transport cholesterol. Now, of course, once we have these back inside the enterocyte, then we will use the enzyme ACAT in the smooth endoplasmic reticulum. ACAT, acylcholesterol, acyltransferase, and ACAT will resynthesize the cholesterol ester, cholesterol with bound fatty acid, covalently coupled fatty acid. Once we reconstruct the ester, then this will be packaged along with triglycerides and, of course, ApoB48 as our scaffold protein to make chylomicrons. The chylomicrons are secreted out the basal surface of the cell and enter the circulation. Now, the endogenous synthesis in the liver, we first have to look at the sources of the substrate for cholesterol synthesis. So let us recall that inside the mitochondrial matrix, we have pyruvate that is converted to acetyl-CoA by the pyruvate dehydrogenase enzyme. The acetyl-CoA, of course, condenses with oxaloacetate in the first step, the entry step of the TCA cycle, to form citrate. The citrate is then transported out of the mitochondria via a transporter, and this citrate in the cytoplasm will be processed further in the next step. But before we look at that, let's look at citrate is still in the TCA cycle. Why is citrate transported out of the mitochondria? Well, recall our next step is converting citrate to isocitrate by the enzyme aconitase. And recall, because this is simply movement of a functional group, a hydroxyl group, this is readily reversible. The next step, however, conversion to alpha-ketoglutarate by the isocitrate dehydrogenase enzyme, this reaction is not reversible. Now, under the conditions, we have to remember our conditions. We are still in the fed state. We are in the liver. Fed state means that we have a lot of energy coming in. So processing energy, processing our molecules, our oxidative molecules, through the TCA cycle is going to significantly raise the NADH concentration inside the mitochondria. This will effectively inhibit isocitrate dehydrogenase. Remember, the dehydrogenases are typically largely regulated by NADH levels. So as NADH levels rise, because we're in a fed state with lots of glucose coming in for oxidation, the isocitrate dehydrogenase reaction will slow and stop. That will build up isocitrate. Once the isocitrate concentration rises, the aconitase enzyme is reversed. We will convert isocitrate to citrate, and then notice this reaction going backwards is also irreversible. It's irreversible because we are using the high energy bond between acetyl-CoA, coenzyme A group, to drive this reaction forward, and that's not reversible. So the concentration of citrate will increase dramatically. Now the citrate is transported out of the mitochondria by an antiporter that will be transporting malate into the cytoplasm. So this is a one-to-one transfer. We'll take a look at this transporter in fatty acid synthesis. Now the citrate will be processed into oxaloacetate and acetyl-CoA in the cytoplasm. The enzyme that performs this reaction is called citrate lyase. So recall, when we were making citrate in the mitochondria, this is citrate synthase. When we're breaking citrate into its respective oxaloacetate and acetyl-CoA partners, it is citrate lyase. Now we're going to talk about acetyl-CoA in the cytoplasm because this can go into two directions. This is a precursor and a substrate for fatty acid synthesis. However, today we're going to talk about cholesterol synthesis, which is the alternative path for acetyl-CoA in the cytoplasm. So the first step of cholesterol synthesis is mediated by the enzyme thiolase, and we are simply going to take two acetyl-CoAs and couple them in a head-to-tail fashion, and we will have acetyl-CoA. Just two acetyl groups conjugated together. The next step is to add a third acetyl-CoA group, and we are going to form hydroxy methyl gluteryl CoA. More familiar, HMG-CoA. The enzyme that performs this reaction is HMG-CoA synthase. Now the next step is probably the most important step in cholesterol synthesis. We are going to convert the HMG-CoA into mevalonate. The enzyme that does this is HMG-CoA reductase. Because we are performing a biosynthetic reaction, we are going to be using the energy stored in NADPH that we made in the pentose phosphate pathway. And the reason that this reaction is so important is because this reaction is the rate-limiting step and the entrance point to cholesterol synthesis. The rest of these reactions are all reversible. This is important when we consider the drugs statins. Statins target HMG-CoA reductase and inhibit their activity as competitive inhibitors. They are competing with the substrate HMG-CoA to block activity. This is a good place to block activity because the HMG-CoA that will begin to build up in concentration by blocking the reductase enzyme has an escape route. Because these reactions are all reversible, we can reverse ourselves back to acetyl-CoA, and the acetyl-CoA doesn't build up in the cytoplasm because, again, the escape route includes moving the acetyl-CoA into fatty acid synthesis. So there are no toxic intermediates because of this drug. Now the next step in cholesterol synthesis, there are several steps involved, and essentially we are going to be making isoprene units. And these isoprenes are 5-carbon structures, high energy structures, because we are going to be now adding these together to start building the structure of cholesterol. We are going to take 6 isoprene units in a series of steps, and we are going to be making the molecule called squalene, which is a 30-carbon linear structure of carbons. Then there are another series, complicated steps. These enzymes are classified as cyclases, and we are going to take the 30-carbon intermediate and we are going to make ring structures. So the cyclase enzymes are ring builders. 4 rings will be added together, will be constructed, and once we have that, we have our final product, cholesterol.